Assalamualaikum warahmatullahi wabarakatuh. Good morning, everybody. How are you today? I hope you'll be fine and keep fighting to start our lesson. First, I want to introduce myself. My name is Rizky Aulia Celestiani. You can call me Aulia. I will be teaching you biology in this semester. Well, before we study, pray basmalah to Allah Subhanahu wa Ta'ala. Bismillahirrahmanirrahim. Now, I want to teach about regulation of expression genes. Let's get started. What is a regulation of expression genes? Gene expression is the process by which the genetic code, the nucleotide sequence of a gene, is used to direct protein synthesis and produce the structures of the cell. Genes that code for amino acid sequence are known as structural genes. For a cell to function properly, necessary proteins must be synthesized at the proper time. All cell control regulate the synthesis of proteins from information encoded in their DNA. The process of turning on a gene to produce RNA and protein is called gene expression. Whether in a simple unicellular organism or a complex multicellular organism, each cell controls when and how its genes are expressed. For this occur, there must be a mechanism to control when a gene is expressed to make RNA and protein. How much of the protein is made and when it is time to stop making that protein because it is no longer needed. Then see the following video to better understand the gene expression. Here is a cell, the basic unit of all living tissue. In the most human cell, there is a structure called the nucleus. The nucleus contains the genome. In human, the genome is split between the 23 parts of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instruction for making proteins. When a gene is switched on, an enzyme called RNA polymerase attacks to the start of the gene. It moves along to DNA making a strand of messenger RNA. Out of three bases in the nucleus, the DNA code determines the order in which the three bases are added to messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be a process. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus in the cytoplasm. Protein structure in the cytoplasm called ribosomes bind to the messenger RNA. Ribosome read the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acids. The transfer RNA molecule carry the amino acid to the ribosome. In the messenger RNA is read 3 bases at the time. As each triple is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein. Okay. it for today. I hope you can understand our material today. Thank you for attention and see you on the next meeting.